High-throughput quantitative data process for genome editing. Fragment analysis for capillary electrophoresis-based study. By fragmentanalysis.com. There are multiple strategies of mutation detection for TALEN, XEN, CRISPR-Cas9 mediated genome editing. Each of them has certain advantages and limitations. For example, sequencing-based analysis is limited by its cost and high throughput capability. EMC-based methods is limited by enzyme capabilities and experiment conditions. T7 endonuclease 1 is proficient at recognizing insertions and deletions of more than 2 bases that are generated by non-homologous end joining activity. However, it does not detect single base insertions or deletions efficiently. Although HRMA is a high-throughput approach, it only provides limited information. There is also limitation on amplicon size because of the resolution requirement. Capillary electrophoresis-based strategies can provide precise, simple, cost-effective, highly sensitive solutions with single base pair resolution. There are no limitations on amplicon size, which can reach thousands of base pairs. They are high-throughput approaches which can be applied for both cell clones, cell pools and whole animal. However, the limitation is automatic quantitative secondary data process specifically required by the genome editing study. Now let us explore more details about C-related approaches. Here are most recent publications, including the methods from enzyme characterization, IDAA, fluorescent PCR and CRISPR-STAT. Here is the summary of oligo design schema from above listed publications. No matter what the difference of the design, they all finally go through C instruments for fragment analysis. Here are some example of data output from C instruments from the listed publications. The ideal case is as the figure from IDAA. There are 3 analysis regions: wild type, deletions and insertions. There is only a single peak in each region. However, in real experimental data, it can be as simple as this case, or be very complicated like this one with multiple peaks in each region. Furthermore, not only classification but also quantification are required for some study. Let us take a look on the general workflow of your data process. You perform experiments, and submit your samples to any core facility or company who can provide fragment analysis service. After you get the output FSA files from C instrument, you can use GenMapper or Peak Scanner to do primary data analysis, including baselining, peak detection and sizing. If you are a new user for data processing or want to learn more tricks of the software, go to fragmentanalysis.com video gallery site. It is important to adjust analysis parameters to maximize your experiment measurement capability. Video PSO4 and PSO5 would be a good help. However, either GenMapper or Peak Scanner can provide secondary data process for genome editing, which include filter, grouping, annotation, quantification, statistical analysis and visualization. This is the bottleneck of high-throughput study. How to extract important biological insights from thousands of rows of data within seconds. No worry. We can help. For the first-time user, please take some time to read our tutorial and watch video FA02. For the simple case like this, please refer to the information from the tutorial and video. Here, we spend some time on a complicated case. For example, for this study, there are multiple peaks around wild-type positions. However, there are 2 distinct fragments in the deletions region. And there are no obvious insertions. For this case, it would be better to design the following analysis set. It represents the following selection criteria. Only choose fragments from DIB since they are FAM-labeled the fragments. In region from 178 to 182 BP, select top 2 fragments to calculate del1 percentage. By this way, it will reduce the chance to lose any potential fragments in this region. Later on we can confirm it by counting fragment number in each region. It should be 1 if only single fragment falls in this region. However, if you are sure that only one fragment in this region is expected or required for percentage calculation, you can input fragment number as 1 here. Apply the same method to design other regions. For wild type, select top 5 fragments in region from 197 to 203 BP for percentage calculation. Here is the demo of some results for genome editing study. In the analysis result page, there is a link results table with fragment number. Click this, you will get additional information for data analysis and interpretation. The results table contains additional columns for fragment number information in each region. You can download the data from this table for further analysis and visualization by any software, such as JMP, Tableau, Spotfire. Several visualization figures are available at fragmentanalysis.com. The figure fragment percentage by sample displays the fragment percentage in stacked column format. The figure fragment number by sample shows the number of fragments in each predefined region. You can choose to display the results for one or multiple regions. For example, it shows here for all samples with fragment signals, the wild type region contains 4 peaks. If you have any questions or challenges, contact fragmentanalysis.com for support and solutions.